So we'll get started. This chapter in the textbook is particularly challenging because it covers a really vast number of different topics that are only integrated sort of loosely. I was originally only going to talk about stem cells and cancer, but I'm going to just give a brief overview of what the book presents about tissue organization, because it is relevant to thinking about cancer and metastasis in particular. And it's not something I know a great deal about, and I don't think you need to know a great deal about to be a functional cell biologist, but I think it's useful to just discuss a few things that we haven't yet gotten to. Before I get started with that, I just want to advertise for those of you who are interested in genetics that there's a very large conference that was supposed to be held in Washington, D.C. this week. That is going to be a virtual conference, and it's free to anyone who wants to attend. So if you're interested in genetics, I would suggest you register for this conference, and you can attend as much or as little of it as you want. There are some undergraduate research presentations that you might find interesting in particular, and you can pick and choose among the topics. Okay, I hear my cat screaming, so I'm going to go let her in. Sorry. I'm back. All right, so tissues. The main point that I want to talk about with tissue organization is that in multicellular animals and organisms in general, there are a number of different types of cells that typically contribute to a tissue, and then tissues can be organized into organs. Okay. I thought I'd solved a lot of my video problems, but I see I've introduced some new ones now. All right, so these are just examples of complex tissues with cells arranged in layers. Those are epithelia, but they're embedded in this material, a lot of which is actually extracellular. It's made of proteins and sugars and other biomolecules that are made inside cells but exported into the extracellular space where it forms a variety of different kinds of extracellular matrix, which can be very rigid, like bone, or soft, or flexible, like tendons, and it gives rise to a lot of the structures that we see in the body. So this is just an example of how a tissue is organized with layers of actual cells that are attached to connective tissues. Some of which, the connective tissues often have cells embedded within it that are creating the extracellular matrix material. And there's adhesion between the cells and the extracellular matrix that organizes the tissue and gives it strength and rigidity. I'm just going to check the chat. Sorry. Okay. So this is just an example of the lumen of the gut, which is a tissue that's used at several points in this chapter to illustrate specific points, which is why I decided to show this particular slide. These are other examples of tissues that are rich in extracellular matrix material, also called connective tissue. So this is bone, and in this case, this preparation was done in a way that most of the cells were sort of extracted, or only residues of the cells were left. So most of this is extracellular material, and you can really see the striations here in part because it's imaged with polarized light microscopy, which is very good at showing kind of the directionality of long fibrous molecules. So bone and skin are made of polypeptides, of proteins that have this sort of coiled, very tensile structure. That's often, in the case of bone, it's intercalated with crystals of, I think, calcium, is it carbonate? I'm not sure, that give it rigidity. In other cases, there's sugar molecules and other modifications that add to the biophysical properties of the material. So the cells are embedded within this material, and they're connected to the extracellular matrix through a variety of connections, between proteins that span the plasma membrane of the cells and the extracellular matrix proteins themselves. So the book points out this one particular kind of disorder that is sometimes occurs it doesn't name it, but I added the name here. This is a very large group of disorders of connective tissue synthesis, where you can have overly stretchy skin if the collagen in your skin is not properly synthesized. Marfan syndrome, I think, is an elastin deficiency. So collagen and elastin are kind of the two major proteins that contribute to extracellular matrix in animals. And so these are different kinds of defects. They're not fatal. In fact, I think Michael Phelps is thought to have Marfan syndrome. It can make you very long and flexible, but it also gives you a distinctive facial appearance. And then I didn't know this until preparing for this lecture, but scurvy, which we don't see very often in modern day, but was a common ailment of sailors who didn't have any fresh fruit to eat, because you need vitamin C to produce the collagen that gives rise to a healthy extracellular matrix. And so sailors who didn't eat limes or some other source of vitamin C would often develop scurvy. Their teeth would fall out due to loss of the tensile strength in their gums and so forth. So, all right, so this is just an illustration of how cells are connected to the extracellular matrix. It's more complex than this. This is just one example, but it shows these proteins called integrins, which span the plasma membrane. They're a dimeric protein, so they have two subunits, here shown as a green and a blue subunit. And they're connected not directly to the collagen fibrils, but to a connector protein called fibronectin that binds to both the integrins and to the collagen. And then on the interior of the cell, the integrins connect to adapter proteins, which then connect them to cytoskeletal structures, particularly the actin cytoskeleton. And the book points out that these integrin molecules have this interesting way of binding to the extracellular matrix, such that they're in an inactive form when they're not connected to the extracellular matrix, but when they bind and when there's force on them, that actually increases the strength of the bonds and induces them to connect to the cytoskeleton. And this kind of molecular bond that gets stronger when you put force on it, it behaves like one of these Chinese finger traps, is called a catch bond, just if you're interested. So in this case, the integrin molecule is actually communicating with both the extracellular matrix and the interior of the cell, and there's interplay between the binding to the extracellular matrix and the cytoskeleton. Okay, so a lot of the tissues in our body are epithelia. They are typically sheet-like arrangements of cells, probably the primordial cell type that gave rise to the earliest single-celled and then multicellular animals were epithelial cells. And they can be arranged in different ways. When the cells are very long and skinny, they're called columnar. When they're flat, they're called squamous. When they're kind of evenly tall and wide, they're called cuboidal. And then they can also be arranged in more heterogeneous arrays like this. But there are 2 points that are made here in the book. One is that the basal, so there are actually 3 points. So they're polarized. They have what's called an apical surface, which is away from the supporting connective tissue. The basal surface, which is connected to that tissue, which is called the basal lamina. And they have, so they're polarized, and I mentioned in a lecture about membrane structure that there are kind of boundaries between the apical and basal, or basolateral, as it's sometimes called, sides of the cell, so that membrane proteins are confined to one layer or the other because of borders or boundaries that are in the membrane. So, yeah, so this just shows that at higher magnification. Here's the apical surface and the basal surface that's rich in connective tissue or extracellular matrix. And these sheets of cells can either be sort of flat, or they can curve and seal up When that happens, these cells tend to take on sort of a wedge-like shape to accommodate the curvature. And by doing that, they can form a variety of tubular or spherical structures. And these just show a couple of examples of like the neural tube and structures in the developing eye in a chicken. And so I'm not going to go through this in any detail. The book goes through this list of different kinds of connections between cells that have different functions. Some of them allow, like gap junctions, allow things to go between small molecules like ions and water to travel between cells. Some provide more of a structural connection where the actin cytoskeleton of one cell is connected to that of an adjacent cell. I'll just mention tight junctions, which are barriers to diffusion of proteins within the membrane, but also they form a seal, so that they actually allow a cell layer to behave as an impermeable barrier to molecules. And you can see that by applying, some kind of fluorescent or dye molecule to one surface. If you apply that to a cell layer that's connected by tight junctions, the dye will be confined to the layer that you apply it to. So if you apply it to the apical side, it will stay on that side. And conversely, if you apply it to the basal side, it'll kind of fill these spaces, but it won't spread to the apical side. And that's just shown here with some kind of tracer molecule. This is an electron micrograph, a transmission EM image, showing some kind of tracer here that does not go through this tight junction into the basal space here. And conversely, here the tracer is applied basally, and it doesn't penetrate the apical side. Okay, so I think the main reason that this whole section on tissue organization and layers and so forth is in this chapter about stem cells and cancer is to highlight the fact that most tissues and all tissues and organs are complex collections of cells, but by talking to each other, the cells control the differentiation pathways that all the cells take, and they control proliferation. So these cells are all different, but they're all kind of talking to each other in a way that maintains homeostasis, so that you have the right number of epidermal cells and keratinocytes and all the other cell types that are present in the skin are present in kind of the right abundance, and they maintain their correct positions within the tissue. This is just an H&E stained section of mammalian skin to illustrate this more in real life. You can see the nuclei again stained with this blue dye, and the eosin is the pink dye that stains the cytoplasm. Okay, so these are all examples of differentiated cells. These cells have taken on specific types of fates and structures that enable them to perform their functions in the skin. But these cells arose from an ancestral cell that was a stem cell. And there are different kinds of stem cells. Kind of the mother of all stem cells is an embryonic stem cell. So this just illustrates the fact that a single cell, an embryonic stem cell, can give rise to an entire organism. So if this is a sea urchin stem cell, it'll give rise to a sea urchin. And if this is a mouse stem cell, it'll give rise to, I think this is a mouse. Meaning that the progeny of this cell, as it divides, have the capacity to become every kind of cell that makes up a sea urchin or a mouse, respectively. And because of that ability to differentiate into all different cell types in an organism, these embryonic stem cells are considered to be totipotent. They can become everything. As they start to produce offspring cells, some of those daughter cells are what's called pluripotent, meaning that they can give rise to a spectrum of different cells, but their ability to give rise to all cells in the body becomes more limited with over the course of development. So this just illustrates that in a little bit more of a schema, where you have, so most stem cells have this kind of asymmetric division where they give rise to daughter cells through mitosis, And, but the fates of those daughter cells are asymmetric. One of them is maintained as a stem cell, whereas the other one becomes more differentiated. So it turns on different transcription factors, and it starts developing in a way that makes it closer to a terminally differentiated cell. But this cell can still be pluripotent, meaning it can still maybe give rise to a spectrum of different terminally differentiated cells. So as cells become terminally differentiated, they stop dividing. They exit the cell cycle. And that's an important part of differentiation. So this just shows another way of looking at this. This is an early embryo, again, from a mouse, or it could be a human. I think this is more of a mouse illustration. And these cells, which are going to become all the tissues in a mouse, can be removed from the early embryo and cultured. When we culture them, we call them embryonic stem cells. And by applying the signals, the growth factors and differentiation signals that trigger differentiation into these different types of cells, we can actually watch these differentiation processes happen in a dish, which is a super powerful tool in cell biology and developmental biology. And this just shows examples of differentiated cells in a dish. So this would be like an embryonic stem cell that, or a neuroblast that has been given the right signals to differentiate into neurons. So now this is a neuron. It's not going to divide again because neurons have exited the cell cycle. But it has it has the morphology of a neuron. It has the signal transduction capabilities of a neuron and so forth. And similarly, these cells are derived from a myoblast progenitor, which can differentiate to become myotubes, which are the components of muscle. So part of why I'm bringing this up is because we used to think that differentiation was a one-way street, really until like the last 20 years. It was sort of dogma that once a stem cell, a pluripotent cell, was induced to differentiate, that was irreversible. You couldn't go back. And you know, that there was this kind of major shakeup in developmental biology in the early 21st century, in which people figured out how to take differentiated cells and de-differentiate them back to a pluripotent state. And this is just an illustration of some of these early experiments where people removed, you know, somewhat differentiated cells. They hadn't yet exited the cell cycle, like fibroblasts, from a mouse. And they found that by just expressing this kind of magic combination of transcription factors that are very important determinants of the stem cell-like fate, that they could actually revert these cells to a de-differentiated state. They could inject them into a mouse embryo, put that in a mouse. And in this case, these cells make up just a small fraction of the total cells in this developing mouse embryo. And so the resulting mouse, well, they're putting this embryo into a pregnant mouse, and her offspring will now be chimeric. They'll contain some cells that are derived from these cells that were removed from this mouse, de-differentiated, and injected into the embryo. And if these cells populate the germline of this chimeric mouse, then that mouse can give rise to a fully, a mouse that is genetically related to this mouse, but not this mouse. So this is now a very powerful tool for developing cells in the lab that have the ability to become essentially any type of cell in the body. And there's enormous interest in this area of stem cells because people are very keen to use this as a way to generate new tissues for regenerative medicine. We're still not like all the way to being able to replace cells in a human body with cells that are derived from the same person but reprogrammed. But that is sort of the goal, is like if you have a defective kidney or need some other kind of transplant, you might be able to take your own cells, reprogram them, get them to develop into the tissue that you need that's damaged, and put it back into your body. But in order to do that, we have to understand not just how to de-differentiate the cells, but also how to in a very orderly way, get these cells to differentiate into the tissue that we're interested in. And so people study in particular, cells of the hematopoietic system, so the blood and immune system, or other kinds of developmental pathways in the hope of being able to replicate those pathways in vitro so that we can create organs that are derived from our own genetic makeup. And that in that way, we can avoid the kind of rejection that often accompanies transplantation because of genetic differences between the donor and the recipient. Okay. So in order to do that, we need to either take human cells and turn them into essentially embryonic cells, which are called induced pluripotent stem cells, or in some cases we may be able to do it by culturing stem cell populations from the tissue of interest. And this slide really illustrates a slightly different point, which is that most tissues that are made of differentiated cells, as I showed, that are arranged in kind of a nice order, have a subset of cells within them that are stem cells, and they are the cells that divide to produce the different kinds of cells in the tissue. This example again is the lumen of the mammalian gut, and because that's a highly acidic environment and it's subject to a lot of environmental insults, these cells turn over constantly, and they are replaced on the order of every 3 to 6 days by cells that arose through division of these dividing stem cells in what's called the crypt, kind of at the bottom of these villi. So these are villi, and the cells that make up the villi also have these little microvilli on top that increase their surface area to allow for nutrient absorption. So yeah, I don't know if there's anything else I should say about this. This just shows that as the cells are dividing, they're also kind of moving upward through this column to repopulate these villi. Okay. So similarly, the cells in the bloodstream, including all of these different types of blood cells, and also including the cells of the immune system, B cells and T cells, are derived from a population of stem cells. The book, for some reason, calls them hemopoietic stem cells, but most people call them hematopoietic stem cells, and I'm not sure why they've used this nomenclature, but they're, I think, equivalent. And so there's a population of hematopoietic stem cells that give rise to all of these different types of cells in the blood and immune system. And there are some of these free in the blood, but the majority of them are found in the bone marrow. And this is just a figure I pasted in to show that there's 2 types of bone marrow. There's yellow bone marrow, which is mostly fat cells, and there's red bone marrow, which is rich in these hematopoietic stem cells or hemopoietic stem cells, and is vascularized. So as these stem cells produce offspring, they can go into these blood vessels and enter the circulatory system. Okay. So in order for cells to keep proliferating, they have to be able to replicate their DNA faithfully, and they have to do something special that we didn't really talk about, which is they have to maintain the ends of their chromosomes. So most cells in the body are mortal. If you take them out of the body and put them in a tissue culture dish and give them nutrients and everything else, they will divide, but over time, the population will shrink because some of those cells will basically get to the end of their replicative life. They will not be able to divide again. And we don't know all the factors that limit the ability of cells to keep dividing. There's more than one. But in human cells, one of the limitations is the absence of telomerase, which is necessary to maintain the ends of the chromosomes. So someone asked, is that red the type of bone marrow used commonly in transplants? Yes. I don't know if they can selectively remove the red marrow or if they take, you know, I don't know actually the details, but that is where the stem cells that, you know, if you have leukemia, that means that there's a defect in your stem cells, your hematopoietic stem cells, and usually one of the treatments that is commonly given for blood and immune disorders is to irradiate a patient so that kills their stem cells, and to take bone marrow from a donor and put it into the recipient, and then basically it's a stem cell transplant. Okay. So we didn't really talk much about telomeres. I think when I described chromosome organization, I mentioned them, and I think Gary may have mentioned them as well. But I mentioned that eukaryotic chromosomes are linear, and the very end of chromosomes is this repetitive sequence called the telomere. The telomere is actually the DNA sequence plus these proteins that bind to the end of the chromosome, collectively called the shelterin complex. And the purpose of this complex is to protect the end from fusing with other chromosomes, and so that the cell does not see this end as a DNA break and respond to it as if it had some kind of DNA damage. But this shelterin complex, which is present in all cell types, cannot replicate the end of the chromosome. For that, you need this complex called the telomerase complex. And the telomerase complex is active in stem cells and also active in many cancer cells, but it gets turned off in differentiating cells in humans. And it's not really known why that is. It's not true in mice. Mice have active telomerase in all their cells, but it is thought that it's actually a way that cells protect themselves from cancer by preventing their own infinite division. And so the reason you need telomerase, if you think about the mechanism of DNA replication, you have when you have a replication fork, the way it works is you have an RNA primer that is put down on the leading strand, and that can synthesize out. But the lagging strand has a problem because when you degrade, you can't go all the way to the very end with the normal mechanism of telomere of DNA replication, because you need the RNA primer to synthesize this segment of DNA, but then there's nothing, you can't finish it because the RNA primer gets degraded, and there's no way to put a primer beyond the end of the chromosome to template this part. So each cell cycle, the telomere gets a little bit shorter. And you can see that in human cells. This is a southern blot where people have taken the chromosomes from human cells at different stages of, so they've taken human fibroblasts that they're growing in culture, and they've collected them at time 0, and then let them replicate for a number of population doublings, taken another sample, and so forth. And by probing this blot with that telomere repeat sequence, what you can see is that the telomeres on average they shrink. So they go, in this case, from an average of several kb down to just a few kb. And when the telomeres get short enough, that triggers an arrest signal. And that is part of why cells hit this replicative limit and start to no longer divide. But what often happens spontaneously in a population of cells is some small population will reactivate telomerase, or they will find a way around not having telomerase. And specifically, they use another mechanism called ALT, for alternative telomere maintenance, to replicate their telomeres and to keep dividing. And so that's what it says here. So often in the course of carcinogenesis, or the beginning of cancer, one of the key things that happens is that cells that are heading towards being cancerous will reactivate telomerase or turn on this alternate mechanism called ALT to maintain their telomeres, and that way they will be able to grow forever and grow and divide forever. So what's illustrated here is that cancer is a progression. Initially, there's some mutation that leads cells to undergo what's called hyperplasia or overgrowth, excessive growth. And that typically arises through loss of the kind of regulation that normally keeps cells nice and ordered within a tissue. So you might, wonder, why is it that an epithelium is just one cell layer thick? And that's because there's something called contact inhibition. When a cell is touching cells on both sides, that typically prevents it from dividing. Even though it has the potential to divide again, it stops dividing. But if a cell loses that kind of stop signal when it touches other cells, then it can undergo this kind of hyperplasia. And then typically so now there's sort of an evolution process where these cells become abundant. And if one of them develops additional mutations that cause it to take on aberrant cell fates and no longer want to stay in this layer, that's called dysplasia, like a change in fate. And if it starts to become invasive, then you have a serious problem, because these cells are no longer just growing and creating a benign tumor. They have the potential to invade adjacent tissue, and eventually they may acquire the ability to penetrate the tissue, enter the bloodstream, and travel to other parts of the body and colonize new sites, which is metastasis. And that is usually what becomes lethal in cancer. Okay, so this is just an illustration of that process. So this is similar to the image I just showed, but so here's a population of epithelial cells, and here's a mass that developed first hyperplasia, and then the ability to invade this connective tissue, which typically involves expression of metalloproteases by these cells that enables them to actually digest the connective tissue. And then the kind of worst case scenario is when these cells gain the ability to penetrate the epithelia that make up the capillaries and the arteries and veins that make up our circulatory system, because that's how they get elsewhere in the body. So that's pretty much what's said here. Yeah, okay. So there's been a couple of major reviews that talk about the hallmarks of cancer cells, and this is just kind of a summary of the different kinds of defects that typically are associated with malignant cancers. And some of them I've already alluded to, but so I've talked about this, that cancer cells tend to lose the normal stop signals that tell them to stop dividing when they're in a tissue adjacent to other cells. I've talked about this, that they have this replicative immortality that often comes through reactivating telomerase, which shouldn't be on in most somatic cells. And then there's some additional features, some of which Gary will talk about later this week. He's going to talk about the nature, so how these kinds of changes happen in cells that gives rise to all of these different properties, and become sort of a feed-forward loop of genome instability, leading to more mutation, leading to acquisition of more of these traits that make cells particularly dangerous. Yeah, I didn't mention this, but most cells also undergo cell death under conditions where their normal environment is disrupted, but cancer cells typically lose the apoptosis response to those kinds of signals as well. So they again become hyperaggressive. And this is the book's kind of summary of essentially this much more complex picture, but it just highlights the fact that there are certain pathways that are mutated in most cancers. And we've talked about a couple of them. So for example, I mentioned in the cell cycle lecture that the most commonly mutated gene in cancer cells is p53, which is a component of the DNA damage response. So not all cancer cells, but many, maybe most cancer cells lose the ability to detect their own DNA damage through mutations in p53 or other DNA damage proteins. A key example actually is BRCA1 and BRCA2, which are named for their association with breast cancer. Those are also DNA repair proteins that, when mutated, cause a much higher incidence of cancers. BRCA1 and BRCA2 mutations are particularly associated with cancers of the reproductive system and breast tissue. It's not always clear why certain kinds of mutations are associated with particular kinds of cancers, but that is often the case. So BRCA1 and BRCA2 are associated with breast cancer, pancreatic cancer, and I think a few others. And the book talks about, I think because it starts out talking about the structure of the intestinal lumen, it also talks about colon cancer, which arises in that tissue. So I'll show that in a second. We also talked about RAS, which is a major G protein involved in G protein coupled receptor signaling, and activating mutations in RAS are, I think, the second most common mutations in cancer after p53 loss of function mutations. So as I said, Gary's going to talk more about the kind of genetic and genomic aspects of cancer and how these mutations arise. But this is just it looks a lot like the non-disjunction and maternal age graph that I showed you. It's similar in the sense that the incidence of cancer in the human population is a very strong function of age. And that is largely because it takes, we think, multiple mutations for cancer to arise in a cell population. And so the chance of that happening increases dramatically over time. And in addition, other things, there are other factors associated with aging that reduce cells' ability to repair themselves and to respond appropriately to extracellular signals. So this is just another kind of way of representing the progression of cancer from a single mutation in one cell to a proliferating group of cells that provides opportunities for additional mutations to occur and to create more havoc and eventually to lead to a very dangerous phenotype. So this is a chart version of the different kinds of things that can happen that can lead to mutations that may get worse as cells age. So defects in replication, repair, cell cycle checkpoints, I've already alluded to p53, mistakes in mitosis, which can also cause like chromosome missegregation and create aneuploidy. There's a lot of controversy about the role of aneuploidy in cancer, as you may be aware, but it is a contributing factor in many cancers. And so abnormal chromosome numbers are associated with many cancers. The controversy is really about whether that's one of the things that starts the process or one of the things that happens kind of along the way. And that's still unresolved in most cases. And this, again, is just a list of key proteins that are mutated in many cancers. And I just mentioned RAS and p53 and BRCA1 and 2. And the other two on this list are beta-catenin, which I did not, I didn't really talk about in this lecture, but it is mentioned in this chapter. It's a key connective protein between adjacent cells, and so it makes mutations in beta-catenin can make cells resistant to growth inhibition due to adjacent cells. Are people with Down syndrome more susceptible to cancer? I don't think so. There are some chromosomal birth defects that can make people more cancer prone, but in general, the aneuploidies that are tolerated to adulthood are not cancer-causing aneuploidies. So Kirsten looked it up and said, yes, they are at higher risk. So I was wrong. Okay. So, and then the book talks mostly about this APC. This is not the APC gene that we talked about before, the anaphase promoting complex. This is something that I can barely pronounce called adenopolyposis coli, and I don't know what that name means, but I'll show you the relevance in the next few slides. There are a couple more questions, sorry. Yeah. So I just want to highlight, and I think Gary will come back and talk more about this, that these genes fall into two major categories. They're either proto-oncogenes or tumor suppressor genes. So proto-oncogenes are genes that can mutate to a form that causes cancer. Like they're still active, but they become typically hyperactive. So in the case of RAS, we talked about mutations that can occur that make RAS essentially active even when there's no G protein coupled receptor receiving a signal from outside the cell. The RAS just thinks there is and is on. You know, it either loses the ability to hydrolyze GTP or just prefers the GTP bound state more than it should. And so it's on too much. So you might want to go and review that. And similarly, I don't really know as much about beta-catenin, but mutations in beta-catenin can also make the protein sort of hyper active. They can prevent degradation and thereby promote proliferation. Then these three are examples of tumor suppressors, and that means that their activity normally prevents cells from becoming cancer-like. But if they're lost, if you lose a tumor suppressor gene, and Gary will talk about how that can happen, then the cell is sort of deprotected and much more prone to undergo the kinds of aberrant pathways that can lead to carcinogenesis. So these are, I mean, all of these proteins play good, important roles in the body, but these typically get mutated in a way where they're hyperactive, and that causes cancer. And these, if their function is lost, cause cancer. So that's the major difference. So the book kind of goes through a particular example of colon cancer, which is often caused by loss of function of this adenomatosis polyposis coli, not anaphase promoting complex, gene. And that can lead to this hyperproliferation. Well, it leads to the formation of these polyps. This is just a cross-section of one of these structures. And this happens, my understanding is that this happens because as the stem cells in the crypt of this intestinal tissue are dividing, that their division is regulated by a signal called Wnt. And Wnt is a common signal that is present in many different differentiation pathways. But sorry, this is probably not ideal. So a Wnt signal is an external signal that normally acts through a G protein coupled receptor to turn on a pathway. In this case, though, this APC, or here, this APC acts as an inhibitor. It destroys beta-catenin, which is a signaling protein, in the absence of signal. And if the APC goes away, then you have too much beta-catenin and too much active transcription of these Wnt targets, which leads to hyperproliferation of the stem cells in the crypt and over, hyperplasia, which is what these polyps are. So initially, these are not terribly dangerous, but because they're sort of ripe for further development into colon cancer. So when you have a colonoscopy, they're looking for these things, and often they can basically cure you of colon cancer by removing these before they have a chance to go farther down the pathway to becoming malignant cancer cells. These by themselves are not malignant, but if they acquire additional mutations, they can be. So this shows sort of the progression of a polyp to a more dangerous state, with the initiating event being loss of this APC tumor suppressor gene. So then you get a polyp, excessive proliferation, And then you can get subsequent mutations, often activation of RAS, which can happen by a single base change in the genome, just a single amino acid change in RAS. And that's not a very frequent event, but if you have a big population of these hyperplastic cells, then the chance of having this occur is greatly increased. So if RAS gets activated or some other proto-oncogene gets activated, that can lead to the formation of a small tumor, and that can progress to a large tumor and then an invasive cancer. And that's when things get really bad, especially when you get metastasis. So it can take a long time, but it can, which is why looking for these polyps is an important part of healthcare, especially once you're like my age. You're supposed to be having a colonoscopy every couple of years. Unfortunately, they're rather unpleasant procedures. So there are other ways to detect potential proliferation of these cells in the colon, which are still being developed.